Hello everyone, this video is in continuation to our previous videos where we were discussing about the chapter reproduction in flowering plants, and we have discussed till development of a dicot embryo. In this video we'll be taking up development of a monocot embryo. We'll do in detail what are the different changes which occur in the development of a monocot embryo. Please watch the video till the end, and because in the end I'll be making a flow chart here so that you can copy that flow chart. It will be very easy for you to learn the whole topic. So starting up with the division of monocot embryo, there are certain differences between division of dicot and a monocot embryo and a cotyledons formed. We'll be finding that in the due course as we move on. So starting up with the diploid structure which is formed first, that is the zygote. This diploid zygote has already been formed inside the embryo sac. The zygote will undergo first mitotic divisions to form a structure having 3 different cells. Here also we'll mark the chalazal and the micropylar end to avoid the confusion that which type of cells are formed towards the which side. Here you can see the drastic addition. In the dicot embryo there were 2 cells formed here. Now here we have the formation of 3 cells. Towards the chalazal end we have terminal cell, in the middle we have the middle cell, and we have the basal cell towards the micropylar end. These are the 3 types of cells. All the 3 different cells will undergo different divisions to form different structures now. So we'll use different colors for all the 3 to demarcate. The first is terminal cell. The terminal cell is going to undergo divisions to form the cotyledonary part or the cotyledon. So the terminal cell will divide to form the cotyledon. Next we have the middle cell. The middle cell is going to undergo further divisions to form many structures, and these many structures are the plumule, the hypocotyle. The plumule will be formed, the radicle will be formed, hypocotyle will be formed, and suspensor will be formed. So we have these divisions here, and what is formed here? Plumule, radicle, hypocotyle, and what will it form? Suspensor. And in the last we have the last cell, the basal cell. It will grow and enlarge to form or to absorb nutrition. Grow and enlarge to absorb nutrition. Now this is the second division. After this we have a structure which is formed, and that structure will appear like this. This is the next division. I'll make it here. This structure will appear like this. We have the basal cell here with a suspensor attached here. Then over this part we have these leaf-like structures with sheets. We have these sheets here. I'll use different colors for both, and we have sheets on the other end as well. So this part of the plumule covered with sheets, plumule covered with sheets is called coleoptile. So this part is the coleoptile. After coleoptile we have the next part that is known as epicotyle. The epicotyle part. This is the epicotyle. After epicotyle we have the cotyledonary node. Then after that, this portion here will form the hypocotyle, and this part at the edge or the micropylar end covered with sheets, this part at the micropylar end covered with sheet, or you can say the radical covered with sheets is called, this part, coleorhiza. This part is known as coleorhiza. And we have the hypocotyle here as already done. This part would be the hypocotyle and this part will be the coleorhiza, the radical covered with sheet. Now what happens? These are the parts which are going to give rise to these structures. The upper part towards the chalazal end, coleoptile, that is the plumule covered with sheets. Then we have the epicotyle, we have the cotyledonary node where the cotyledon will be attached. I'll make the cotyledon here. The cotyledon is attached here at the cotyledonary node. This is the cotyledon. Now we have a speciality here. The cotyledon in case of monocots, it's a very thin papery structure which is known as scutellum. It is thin papery structure and it does not provide any nourishment to the developing embryo. I'll tell you how that nourishment comes, that we'll tell you later. But this is the cotyledon attached to it. We have the basal cell and the suspensor here. Basal cell, suspensor, and these will degenerate. These parts will degenerate and these will further develop. Now what structure do we get? If we observe this structure, we'll have this cotyledon attached to this part, and here we have the coleoptile and this side we have the radical and this is the cotyledon. So this will be the cotyledon, this is coleoptile, this is coleorhiza. And how do we see it in a monocot seed? How do we observe it? That's something important. Here only I'll tell you that how nourishment is provided. Now if we see a monocotyledonary seed, we have this outer portion which is the proteinaceous part or the aleuron layer, sorry. The outer part, the proteinaceous part or the aleuron layer. Inside we have starchy endosperm present. Okay? Ab, the part which we have drawn here will be present here at the basal part, and how will it appear? The same, we have the cotyledon here attached to the basal structure. This is the embryo and the cotyledon. So I'll just label, this is the embryo. Now how does this embryo get nourishment? This starchy endosperm will digest in future and will pass its nourishment inside the embryo through the cotyledon. I'll just write it here. Endosperm digests and pass the nutrition to the embryo through the cotyledon. This is the way the embryo starts developing or develops in a monocot seed. We'll recapitulate once again. The zygote undergoes mitotic division to form terminal, middle and basal cell. Terminal cell divides to form the cotyledon. Basal cell form plumule, radical, hypocotyle and suspensor. And the last part, the basal cell grows and enlarges to absorb nutrition. After the further divisions, we have this structure which is formed. Now the parts which we need to learn here are that the plumule part which is covered with sheets in case of a monocot is known as a coleoptile. The radical part which is covered with sheets in a monocot seed is known as coleorhiza. We have a hypocotyle. A cotyledonary node is the part where the cotyledon is attached to the embryo. We have a hypocotyle here. The basal part and the suspensor will degenerate in future. They will be there inside the seed, develop inside a monocotyledonary seed. And how will the developing embryo get nutrition? The starchy endosperm of the monocot seed will digest and pass its nutrition to the embryo through the cotyledon. And something important here is that we have a very thin and papery cotyledon in case of monocots known as a scutellum, which does not provide nutrition, but yes, it is there because the nutrition from the endosperm will enter inside the embryo through the scutellum. So this is all about the development of a monocot embryo. From our next video, we'll start with the formation of fruits. So please keep watching, liking, sharing, subscribing. Thank you.